Hi there, I'm Mrs. Gay. We're now on section 4.5. Okay. This section is a preview of coming attractions, even though we did it in reverse order. Okay. This is looking at the structure of membranes, and when we look at this little section, it seems really obvious. And in fact, we saw this little phospholipid diagram, the gray and the yellow diagram, and we also saw 4.5b diagram way back when we were studying lipids. Both of those diagrams appeared in chapter 3. So, phospholipid bilayer, okay, made out of phospholipids, 2 fatty acid tails, which are hydrophobic, nonpolar. They could be saturated, they could be unsaturated, or polyunsaturated for that matter, right? And instead of the third fatty acid, there's a phosphate group, which is hydrophilic because it's polar. So the molecule is said to be amphipathic. Amphi meaning both. So it has both polar and nonpolar characteristics. That word is actually not in your book. Okay? And so you see that we have the phospholipid bilayer so that the hydrophobic regions can be tucked away together on the inside. The hydrophilic regions are pointing away to the outside. And it's a fluid mosaic model because those phospholipids can move around between each other, and then we have proteins embedded in the membrane. Some of those proteins are just surface proteins. Some of those proteins are just stuck on the inside of the cell membrane, and other proteins actually pass all the way through. So, I've always wanted to do this. You know how Nilsson takes everybody down to the pool for the Cardboard Classic? I would really like to take everyone down to the pool to make a phospholipid bilayer model. And so what I would do, and my problem is I don't have enough ping pong balls. So what I would do would take barrels and barrels and barrels of ping pong balls, and I would just, we could only see the top part of the fluid mosaic membrane, but I would just cover the whole surface of the swimming pool with ping pong balls. And can you imagine if there were little waves, the ping pong balls would sort of move, right? And then I'd lay a raft, a small raft, on top of the ping pong balls. And that would represent a protein on the external part of the membrane. And then I could put something that would float under the ping pong balls, which would represent a protein on the inside of the phospholipid bilayer. And then I'd take a big old inner tube and throw it in. And that would be a protein that goes all the way through. And it could be like an aquaporin, a pore. And then you could jump through the inner tube into the inside and outside. Yeah, so water could pass through it. So it could be like a channel protein. Ooh. A protein that goes all the way through the phospholipid bilayer is called an integral protein or an integrin. Okay.
This is all later.
This is all, yes, integral or integrin. This is all later as well in the first part of chapter 5. Okay.
Wait, an integrin goes all the way through, but isn't a channel.
It could be a channel, it may not be a channel. Okay. The proteins actually do many things. Okay. Now, so we move on then to start looking, but most of a eukaryotic cell is sort of membrane bound. So the first thing we have is that we're subdividing the cell into organelles that manufacture things and break things down. Oh gosh, anabolic and catabolic again. Making things and breaking things down. Dehydration synthesis, hydrolysis. Okay? And so, if we want to make stuff, we need directions. Okay? I am knitting Norwegian sweaters. I'm dead serious. Last weekend I retaught myself how to knit. I used to know a long time ago. But do you think I could have figured out how to knit all by myself? Maybe eventually. But really, what I needed were directions that told me how to cast on this, get the yarn on the needle in the first place, and what the difference between knitting and purling and how many stitches and how to knit with 2 different colors, right? And then I needed directions. If I'm going to knit a Norwegian sweater, I need really cool Norwegian patterns in my Norwegian sweater, right? And so I needed directions. So no directions? How do you think my mitten would look? Not so good, and it probably wouldn't be very functional. So directions are key. The directions for making everything found in the nucleus. Okay? And so some of the key words that they have in this section. The nucleus is surrounded by a nuclear envelope. Notice it's a double layer. Okay? So there's 2 membranes that form the nuclear envelope. There are nuclear pores. Now notice that in your textbook, they look like the inner tube floating in the ping pong balls, right? Remember the cellular landscape? Is it really a hole? No. It's more like sort of a loose mesh network that you have to get through, right? I think it was green in the cellular landscape, if I remember correctly. The nuclear pore, because it wasn't actually a hole.
Yeah, it was green.
Yep, yep. And so you can look at it back in the classroom if you need to remember, right? Inside the nucleus is the DNA. There are many, many, many names for DNA. If the DNA is all unwound, it's called chromatin.
Unwound DNA.
Unwound DNA. So think about a spool of thread. Chromatin. So if I took my spool of thread and I totally unwound it, I'd have this giant pile of loose thread. That would be chromatin. If I wind the thread back up on the spool, it's going to be more protected, it's less likely to get broken. At that point, I call it a chromosome. Preview of coming attractions. If I've made a copy of that spool of thread, my 2 spools of threads are called sister chromatids. So we have chromatin, chromosome, chromatid. Okay, does that make sense? So all of these words are referring to the DNA in sort of different packaging. We also have the nucleolus. Okay, that's where we synthesize ribosomal RNA. And ribosomal RNA and proteins make up the ribosomes. So sometimes the nucleolus is sometimes called the site of ribosome synthesis. It doesn't actually like totally make the ribosomes there, but it actually produces the ribosomal RNA.
And that's all it does.
Okay?
Is the nucleus inside the nucleus?
Yes, the nucleolus is inside the nucleus.
And its main function is synthesizing
Ribosomal RNA. Okay, so we bring proteins. Proteins actually get made out in the cytoplasm. They get brought into the nucleus through the nuclear pore, and then they get assembled with RNA, ribosomal RNA, and we make the subunits of the ribosome and ship them back out. Okay? So, if we talk about the ribosomes, which get initially sort of built in the nucleolus, ribosomes are the sites of protein synthesis. There is a large subunit and a small subunit. If ribosomes can be floating around in the cytoplasm, in which case they are free ribosomes. Other ribosomes are bound to the endoplasmic reticulum. Okay? So we can find them in 2 places. Most of the proteins that are made on free ribosomes stay in the cytoplasm, and then they function inside the cell. Whereas often, proteins that are made on the endoplasmic reticulum get packaged and shipped to other places outside the cell.
The free ones don't.
But the free ones generally produce proteins that stay inside the cell. Okay? Now, in section, and I'm going to stop there because then we're going to continue on with the endomembrane system in the next section.